Assalamu alaikum everyone. Today we are going to talk about cementogenesis again. In the last lecture we covered the cementogenesis for primary cementum, and today we are going to talk about the cementogenesis for secondary cementum. Secondary cementum is also called the cellular intrinsic fiber cementum, and it is present from the middle third till the apical third of the root, and its function is adaptation. So if you can recall the function and location of primary cementum, you would know that primary cementum was present from the cervical margin till the apical third, and this secondary cementum is present from the middle third till the apical third. So there is a difference in location between these two types of cementum. The primary cementum was able to anchor the tooth in the socket, whereas the secondary cementum has no role in anchorage. However, it only helps in adaptation. So by function, these two types of cementum have different roles, and their locations also differ. So secondary cementum starts forming once half of the root has formed. What does that mean? Once the root has formed till the middle third, only then would secondary cementum start forming. Obviously that makes sense because that is the location of the secondary cementum. Just like primary cementum, secondary cementum is also deposited on the unmineralized root dentine surface near the advancing root edge after the half of the root has been formed till the middle third. This type of cementum forms rapidly and is less mineralized as compared to the primary cementum. However, most of the development is similar as that was for the primary cementum. The difference is that in the secondary cementum, the rate of formation is quite high, and this type of cementum is slightly less mineralized as compared to the primary cementum. The second difference is that this type of cementum has cellular bodies entrapped within its matrix. That is why it is called the cellular variety. And the collagen fibers within this type of cementum are entirely coming from the cementoblasts themselves, so they are called the intrinsic fibers. That is how this name cellular intrinsic fiber cementum for secondary cementum originates. Unlike primary cementum, in the secondary cementum, a layer of unmineralized matrix called the cementoid is present on the surface of mineralized cementum. Now, why do we have this unmineralized cementoid present on the secondary cementum surface? Because this presence of this cementoid shows that the secondary cementum does not start mineralizing right after it has deposited. What does that mean? That just like dentine, the secondary cementum has a lag before it starts mineralizing. That is why a layer of unmineralized matrix is always present between the mineralization front and the mineralized cementum, just like the predentine and osteoid of the bone. However, unlike these two varieties of unmineralized dentine and unmineralized bone, this unmineralized cementum, which is called the cementoid, is not very regular, and it is not an easily discernible layer on the cementum. इस बात का क्या मतलब है कि आपको आसानी से ये वहाँ पर नज़र नहीं आती, जबकि डेंटिन के अंदर आपको प्री डेंटिन की एक visible, easily recognizable layer identify आप कर सकते हैं. Okay. So, the first phase of secondary cementum deposition is quite rapid, and the collagen fibrils produced by the cementoblasts during this phase are haphazardly arranged with most of the fiber bundles oriented parallel to the root surface. अब जो ये टाइप ऑफ सीमेंटम है इसमें सारा कुछ सीमेंटोब्लास्ट खुद सिक्रीट करें. The cementoblasts are secreting the collagen fibrils and they are secreting the non-collagenous matrix proteins, unlike the primary cementum where the fiber source was extrinsic. The fibers were coming from the PDL and they were perpendicular to the root surface. So there's another difference here. One, that the collagen fibrils are being produced by the cementoblasts themselves, so they have the intrinsic origin and they are arranged parallel to the root surface. Cellular cementum is, when it is depositing, because it is depositing in the first phase, it is depositing quite rapidly. So some of the cellular cementum also deposits around the PDL ligament fibers, and hence the PDL fibers become entrapped, not all of them, some of them, they become entrapped in the cementum and they're partially mineralized when the cementum mineralizes. And this happens only in some parts of the apical thirds of the root and the interradicular regions, not all over. And this type of cementum where the PDL fibers are embedded in the cementum and mineralized along with the cellular cementum variety gives rise to the third type of cementum called the mixed fiber cementum. If you remember, the mixed fiber cementum was present on the apical thirds of the root and in the interradicular regions only. So this only happens in the interradicular regions and the apical third of the root. So the intrinsic fibers in the secondary cementum run parallel to the root surface, whereas the extrinsic fibers in the primary cementum run perpendicular to the root surface. The intrinsic fibers are uniformly mineralized in the secondary cementum, whereas the extrinsic fibers are not uniformly mineralized. They are variably mineralized. What does that mean? That they have a central core which is unmineralized, and then they have layers of the outer layers are more mineralized as compared to the core of the extrinsic fiber. So this is another difference between the secondary and the primary cementum. So till now we've talked about many differences, starting from the location of both these types of cementum, the function of both these types of cementum, the matrix of secondary has cells entrapped in it and its collagen fiber source is intrinsic, whereas for primary there are no cell bodies and the source of the fibers is extrinsic. The fibers in secondary are oriented parallel to the root surface and the fibers in the primary are perpendicular to the root surface. The intrinsic fibers in the secondary are uniformly mineralized, whereas the fibers in the primary are variably mineralized, having a unmineralized core. Other than these differences, there are many other differences between the secondary and the primary cementum. The cells that produce these two types of cementum are phenotypically different from each other. However, both the types are cementoblasts, but they have slight phenotypical differences. Within the secondary cementum, entrapped cell bodies called the cementocytes are clearly visible. The secondary cementum has a minor role in tooth anchorage or attachment. It is only involved in adaptation. That is why it is usually entirely absent from the single rooted teeth like incisors and canines, and in these teeth, primary cementum is the predominant type of cementum. However, secondary cementum is only confined to the apical and the interradicular regions of these teeth. Primary cementum covers most of the times entire root length, and sometimes 2/3 from the cervical till the middle third, and then from the middle third till the apical third is the secondary cementum. So these are the differences between the primary and the secondary cementum. It is very important to be able to differentiate between these two types. I'm going to show you a figure of the secondary cementum, a section of the secondary cementum, and I will tell you how you identify the secondary cementum on a section, and that is a very important OSCE question. This figure here shows the section of the root this region here is the why am I unable to use my pointer? Okay, so this here is the dentine. This granular layer that you can see here is the granular layer of tomes. If you remember, granular layer of tomes was the outermost layer of root dentine right beneath the cementum. And here you can see the acellular extrinsic fiber cementum present till the middle third of the root, and then from the middle third till the apical third is all secondary cementum or the cellular intrinsic fiber cementum. This figure is the histological section through the secondary cementum. What do you identify? What do you see here? The first thing that you can see here is this cell, which is the cementum producing cell, the cementoblast. Here you can see unmineralized, slightly small, not small, like a very thin, a few micrometers wide and not very regular layer. This is an unmineralized layer. कैसे पता चला ये unmineralized है? Because it is less, it is staining less as compared to the rest of the matrix. So this is the unmineralized cementum. This whole thing is the unmineralized cementum called the cementoid. This is the rest of your cementum matrix, and if you closely see, you would be able to appreciate the cell body present within the matrix. साइड वाला सारा matrix आपको देखिए mineralized है. कैसे पता चल रहा है? Because there's a lot of collagen and there's a lot of mineral dark color का stain हुआ हुआ है. ये जो आपको नज़र आ रहा है यहाँ पर, this cell body is the entrapped cementoblast, and once the cementoblast gets entrapped within its own matrix, it is called the cementocyte. And the cementocyte, just like the osteocyte, is present in a house within the matrix, and that house is called the lacunae. This boundary or this wall is the house for this cell within this matrix, and this house is called the lacunae. This cementocyte has numerous cellular processes extending into tube-like extensions of the lacunae. These tube-like extensions of the lacunae are called the canalicles. And the cytoplasmic processes of the cementocyte, they reside within these canalicles, and they are called the cytoplasmic processes of the cementocyte. Why does the cementocyte develop these processes? Because now the cementocyte depends on its nutrition on diffusion. Where is the diffusion occurring from? Rest of the matrix is mineralized and there is no source of nutrition here. So these cells, they get their nutrition from the adjacent PDL. And the more the distance of the cell from the PDL, obviously the less are the chances of efficient diffusion. And obviously that would mean that less is the amount of nutrition that the cell gets. So as the cementum becomes thicker and the cementocytes become embedded deep into the layers, the cementocytes in the deeper layers, they would get less nutrition because they are far away from PDL, and diffusion is difficult when the distance increases, and thus those cells that are present in the deeper layers, they lose their vitality and their organelles with time because they're not getting nutrition to maintain their organelles and maintain their cells. So this is what happens with the rest of the matrix of the secondary cementum. This is a very important figure, and you should be able to identify which type of cementum it is by just seeing this figure. I think I've missed a slide. Yep, we have not talked about. Have we? Oh, yeah, we haven't. Okay, so as the cementum deposition increases in the secondary cementum, as more and more secondary cementum deposits, because it is depositing at a very faster rate, what happens is the cementoblasts that are secreting this cementum, they get entrapped within their own secretions. And these entrapped bodies, once they get entrapped in the matrix, their cellular sensory activity reduces, and now they are called the cementocytes. And within the matrix, they are residing in houses called the lacunae. These cementocytes, they are not arranged regularly throughout the cementum matrix. They are haphazardly arranged. However, they do resemble the osteocytes of the bone, but the osteocytes of the bone are very regularly arranged within the bone matrix. These cementocytes, they do not have a regular arrangement. However, they do have processes just like the processes of the osteocytes that extend into the canalicles, but these processes do not reach the surface. The cementocytes entrapped within the matrix, they attain their nourishment by diffusion from the PDL. So when the distance of the cell increases from the PDL, that particular cell would get less nourishment because diffusion decreases. Efficient diffusion decreases as the distance decreases. So the cementocytes in the deeper layers, they become eventually non-vital because they are not getting enough nutrition, and they start lacking their cell organelles. And this is how you define, this is how you would be able to identify the secondary cementum. This is all about the secondary cementogenesis. The secondary cementogenesis occurs till half of the root has been formed, and from there it continues till the apex of the root. The secondary cementogenesis is just like primary cementogenesis, but the difference is that it is occurring at a very high rate as compared to the primary cementogenesis, and it is less mineralized as compared to the primary cementum. And then there are loads of differences between the primary and the secondary cementum. Some of them being, the secondary cementum has cellular entrapments within its own matrix because the cementum is developing at a very high rate, Because of that, the cementoblasts get entrapped within their own matrix, and then they become cementocytes, and they're present in lacunae there. They have cellular processes that extend into the canalicles of the lacunae, and these cells attain their nourishment by diffusion through the PDL from the PDL. As the distance of these cells increases from the PDL space, the nutrition diffusion becomes less efficient, and that results in the non-vitality of the cells in the deeper layer. The primary cementum has extrinsic fibers coming from the PDL, and they are perpendicular to the root surface, whereas the secondary cementum has fibers coming from cementoblasts. They are entirely intrinsic in origin, and they are parallel to the root surface. The intrinsic fibers are uniformly mineralized. The extrinsic fibers are not very regularly mineralized, uniformly mineralized. They have a central unmineralized core. And what else? The cells producing both these types are cementoblasts, but the two types of cementoblasts producing these two types of cementum are phenotypically different. The secondary cementum has no role in the anchorage of the tooth. That is why it is not present in the single rooted teeth. And in the single rooted teeth, the major predominant type of cementum is only primary cementum. In the secondary cementum, a layer of unmineralized matrix is visible at the mineralization front, and that is called the cementoid. Now we are left with the fourth type of cementum, the acellular afibrillar cementum. This acellular afibrillar cementum is a mineralized matrix which has a texture very similar to the bulk of the primary cementum. However, the difference is that this type of cementum does not have any collagen in it. As the name says, afibrillar. It does not have any collagen, and hence it has no role in tooth attachment or adaptation. It in fact has no role at all. It is present over enamel sometimes and dentine close to the cemento-enamel junction in the cervical area of the tooth, and there it is present in patches or spurs. मतलब छोटा-छोटा कहीं ना कहीं afibrillar acellular cementum पड़ी होगी, and the cells responsible for the production of this type of cementum are not known. This is all about cementogenesis. We just mentioned a new type of junction called the cemento-enamel junction. Till now, we've talked about many other junctions, starting from the cemento-dentinal junction, which was between cementum and dentine in the crown. Then we talked about the the first one was the cemento-enamel junction, dentino-enamel junction. Dentino-enamel junction was present in the crown between dentine and enamel. Then we talked about the cemento-dentinal junction. Cemento-dentinal junction was present between the cementum and dentine, and it is only present in the root because the cementum is only present in the root. And where was it present? It was forming at the area of the fibrous fringe, if you remember. And now we're talking about the cemento-enamel junction. By definition, cemento-enamel junction is the junction between cementum from the root and enamel from the crown. So where is this junction forming? This junction is forming at the neck of the tooth where the crown meets the root. So somewhere here, where the enamel from the crown is tapering on the cervical margin, and the cementum from the root is also tapering at the cervical margin. So this cemento-enamel junction is present around the neck of the tooth where the crown meets the root. This type of junction is of various types. In about 30% of the human teeth, cementum and enamel meet as a butt joint, forming a distinct cemento-enamel junction at the cervical margin. What does butt joint mean? Butt joint means that the enamel from the crown and the cementum from the root are meeting edge to edge. If I draw this here, if this is the enamel and this red thing is the cementum, this is how they meet edge to edge. This type of joint is called a butt joint. This occurs in 30% of the cases. As you can see here, the enamel from the crown meets the cementum from the root at an edge. There's no gap in between, or there's no overlapping happening, so this is called a butt joint. This happens in 30% of the human teeth. In about 10% of the cases, the cementum and enamel do not meet and there is a gap in between. And as a result of that gap, from that gap, the root dentine is exposed. And this exposure of the dentine leads to sensitivity at this site. And this site is then more vulnerable to caries because there is a gap there. So if I draw this on a paper, okay, it would look like this. Enamel from the crown and cementum from the root, and if you can see, they do not meet at all. And because they're not meeting, the structure that is exposed because of this is dentine, and this exposed dentine is vulnerable to caries and sensitivity. This type of joint or junction is present in about 10% of the cases. In majority of the cases, about 60%, the cementum and enamel meet such that there is an overlap between these two tissues, and mostly the cementum overlaps the enamel. As you can see here, this yellow thing is enamel and this blue thing is cementum, and at the cervical margin the cementum is covering the enamel, it is overlapping, it is coming over the enamel. This type of junction is present about in 60% of the cases. If I draw this here, you have enamel from the crown and cementum from the root overlapping the enamel. So this type of junction is present in 60% of the cases. This type of junction is present in about 10% of the cases, and this is present in about 30% of the cases. These percentages are very important to remember. If you trace, like if you circumferentially go around the neck of the tooth, the chances are quite high that you might form all of these three varieties on a single tooth. It could be that at one point the cementum and enamel are meeting edge to edge. At one point they're not meeting at all. At one point there could be an overlap, so that chances are that you could see all these three forms on a single tooth. So this was all about the cemento-enamel junction. Cemento-enamel junction, its definition, varieties, and the figures for these varieties are very important SAQ question. Then I'm going to talk about the incremental lines of Salter. Incremental lines of Salter पिछले लेक्चर में भी मैंने डिटेल में बताई थी. The incremental lines of Cementum are called the incremental lines of Salter. They're present in both cellular and acellular Cementum, and they indicate that these both type of Cementum have a periodic formation. As I said, when I was talking about the primary cementum, I mentioned that the incremental lines of Salter are highly mineralized areas in the cementum, and in those regions the collagen is less and the ground substance is more, and the orientation of these incremental lines is parallel to the root surface. The cellular cementum is forming at a faster rate as compared to the acellular cementum, and hence the incremental lines in that cementum are far apart than in the acellular cementum. This is the orientation of the incremental lines of Salter, parallel to the root surface. This is all about cementum. I would want you guys to make an assignment, a handwritten note where you enlist the incremental lines of enamel, the incremental lines of dentine, and the incremental lines of cementum. So I would want that you make a note enlisting the incremental lines of these three hard tissues of the tooth, and another note enlisting the chemical composition of these three hard tissues of the tooth, these enamel, dentine and cementum. दो सवाल हैं. Enlist the incremental lines of enamel, cementum and dentine. Enlist or tabulate the chemical composition of enamel, cementum and dentine, and the time of submission of these is Monday before 12:45. Thank you very much. This is all about cementum. We are going to talk about alveolar bone in the next lecture. Allah Hafiz.